We will now do a test for phenols. Now, phenol contains an aromatic ring with an OH group on the aromatic ring. Many phenolic compounds react with iron chloride in order to produce a highly colored product. In the presence of phenols, the yellow color of the iron chloride will turn into a violet color. What we've done is we placed some solid samples, for example, commercial aspirin, inside a test tube. We will now add 2 milliliters of ethanol, and then we will add our iron chloride to the sample. Remember, if the iron chloride turns purple, it is testing positive for the presence of phenol. We will take 1 to 3 drops of 1% ferric chloride solution. You will notice that the solution stayed a yellow color. We will now test our experimentally made acetyl salicylic acid. Again, we will add 2 milliliters of ethanol. Once we add the 2 milliliters of ethanol, we have our experimental sample inside our ethanol. And again, we will place 3 drops of 1% ferric chloride. You will note that the solution remained a yellow color. We are now going to test salicylic acid, and if you remember, salicylic acid was one of our reactants in our reaction. Again, we'll place 2 milliliters of ethanol to the test tube. Now we will add our 1% ferric chloride solution. Again, we will add 3 drops. You will note a very vibrant violet color. The last thing we are going to test is methyl salicylate, which is also known as oil of wintergreen. Now it is a liquid, so we've added a few drops to the test tube. Again, we will add 2 milliliters of ethanol to our sample. And as we did with the other 3, we will add 3 drops of 1% ferric chloride. You will note a very vibrant violet color.